Hi all, welcome to my channel. Before discussing the histology proper, we will know about the neurons present in the cerebrum. As we know, cerebrum has six layers. The neocerebrum has six layers. So the basic four types of cells will be present there. So the first is the pyramidal cells. As the name itself says, it's like a pyramid. The apex is pointing towards the surface, whereas the base towards the white matter. A large apical dendrite arises from the apex and extends towards the superficial surface, and it divides horizontally also. Numerous basal dendrites arise from the basal angles and they extend horizontally. The axon arises from the base of the pyramid and forms the efferent, projection, association or commissural fibers. Next is the granular cells, also called stellate neurons. They're relatively small multipolar neurons. Their dendrites, they ramify within the lamina gray matter there, and they form synaptic connections with the pyramidal cells. In the axon, they branch there itself and they form synaptic connections with the dendrites of the pyramids. Next is the horizontal cells of Kahl. It is not Kajal, J is silent there. Horizontal cells of Kahl present in the only outermost layer, horizontally present. They also branch with the apical dendrites of the pyramid and other neurons. The fusiform cells are the modified stellate neurons and give rise to the dendrites and axons which extend vertically from opposite poles. The cells of Martinotti, which are multipolar small neurons located in most layers of the cortex, but more so in the sixth layer. They have many dendrites which ramify nearby small dendrites. The axon, which move towards the superficial surface, run horizontally there and make synapses with the pyramidal dendrites and the horizontal cells of Kahl. So these are the basic cells. If you know the structures of these cells, then it's easy to draw the histology of cerebrum. So in the circle, first shade the background, the connective tissue and all. Don't leave it white. So first we'll shade. So the horizontal lamination, there is no exact demarcation between each layers, and each layers will differ in different places. Like in granular cortex, agranular cortex, like in the motor areas, the pyramidal cell layers will be more thick, whereas in the sensory areas, the granular cells will be more thick. So the thickness of each layer differs in different places. Usually the cerebrum is covered by a pia mater, some connective tissue cells. Then the outermost layer, we have only the horizontal cells of Kahl and the dendrites ramifying there. So just write the fibers and very few cells in the first layer. The second is the outer granular layer. It is very difficult to notice this cell bodies. So just keep the nuclei. That's the way it is called granular, because a lot of neurons, the nuclei of these neurons will see as a granules like. It is not that granular layer you don't have pyramidal cells. We have some pyramidal cells. The size of the pyramidal cells will vary. Towards the superficial part, we have very small pyramidal cells. So in the second layer, write more of granular cells, the nuclei of the granular neurons, and very few pyramidal cells which are of very small size. Don't forget to keep the nuclei for the pyramidal cells. Then the third layer, outer pyramidal layer, we will keep little bigger pyramidal cells compared to the pyramidal cells in the second layer. The size keep on increasing as it goes towards the white matter. So keep the nuclei. Even the granular cells will be present in the pyramidal layer. So keep some granular neuron nuclei. And also the dendrites, the ramification, the fibers, the dendrites, axon and all, can just write it in a from the apical dendrite, basolateral and all those things you can show there. And then the fourth layer is again the granular layers. So granules should be more. So write plenty of granules and very few pyramidal cells. Here the pyramidal cells of medium size. So little bigger than the pyramidal cells we have wrote in the third layer. So plenty of granule cells, that's why it's granular layer. Little bigger pyramidal cells. Keep the nuclei and show some fibers which represent the projection fibers also, and also the axon and dendrites which are ramifying within there. So one more important, Towards the inner part of the fourth layer, we have a horizontal striation, that is the outer band of bilateral. It's more prominent in the visual cortex. That's why it's also called striate cortex. In freshly cut specimen, we can see the horizontal striations. It's called band of Gennari. So then the fifth layer, name itself says inner pyramidal cells. We have to write more of pyramidal cells and very few granular cells. Here the pyramidal cells are the largest one. They're also called the large pyramidal cells or the Betz cells. BETZ, Betz cells. These are the one which forms the projection fibers. Keep the nuclei in the center, few granular cells, nuclei also. Make sure you write the fibers, the apical dendrites, basolateral dendrites, and the axon coming there. In the outer part of the fifth layer, we have inner band of bilagia. These are the tangential fibers seen in the outer area of the fifth layer. So this is the fifth layer, inner pyramidal cell layer. So these are the axons of these pyramidal cells layer. So because of these axons of these pyramidal cells and the incoming pleomorphic, the projection of fibers, the white matter from the white matter, so in the sixth layer we have all the cells arranged vertically. So their pleomorphic layer vertically arranged. You have cells of Martinotti present there and the fusiform cells present there. They're little smaller than the pyramidal cells. Keep the nuclei for them and few fibers, the ramifying neurons. The dendrites and axons of the neurons ramifying. Then the last, the white matter, you have just axons, incoming and outgoing axons, and few nuclei which gives the myelin sheath for it. So this is the histology of cerebrum. If you know the neuron type, easy to draw the diagram. This is the labeled one. Thank you for watching.